Dear students, I welcome you all to the 15th session of Reproductive Biology and Developmental Biology paper prescribed by the Mangalore University for 6th semester BSc Zoology. In this session, we shall study about the structure of sperm and egg of an amphibian and the chick. The example that we have selected here for amphibian is the frog. So now let us first look into the structure of a mature spermatozoa of frog. A mature frog sperm is microscopic and elongated, is about 0.03 millimeter in length. So this is how the frog spermatozoa looks under microscope. As usual, as the other spermatozoa, the frog spermatozoa is also composed of an acrosome, head, body, and the tail. So at the anteriormost position, it is composed of an acrosome, and this acrosome is filled with a lot of enzymes that are responsible for the digestion of the plasma membrane of the oocyte once the fertilization is going on. Along with the acrosome, a cylindrical head which contains nucleus and a short middle piece containing centrioles and mitochondria, and a much elongated posterior motile tail terminating in a flagellum. So this spermatozoa frog is a very simple structure composed of 4 different parts: the acrosome, head, body, and the tail. Immediately after the acrosome, you can see a cylindrical head which contains a haploid nucleus or the male nucleus. And this part, the middle piece, consists of the centrioles that are very important for the formation of flagellum, and the mitochondria, which are responsible for generation of energy for the flagellum to produce the movement. The acrosome is an apical cap-like structure and is modified form of lysosome, which produces a chemical called lysines to dissolve the egg membranes. Thus, the function of acrosome is to penetrate the wall of egg during fertilization. The head is elongated, round in structure with nucleus of its own size. The body is main part and consists of cytoplasm and cell organelles with spirally coiled mitochondria. The tail of the frog spermatozoa is the cytoplasmic extension of the body, and it makes the sperm motile or migratory. So all the locomotory force is generated by this flagellum, so that the sperm always produces a wriggling forward movement. If you look at the structure of an unfertilized ovum or egg of the frog, it looks like this. So this is how it looks under a simple microscope. The egg of frog is about 2 millimeters in diameter at the time of ovulation. It is surrounded by two accessory egg membranes in addition to the plasma membrane. Just outside the plasma membrane is a non-living transparent membrane called vitelline membrane developed by the ovum itself. So you can see here there is a membrane called vitelline membrane. And this vitelline membrane is the one which is present just outside the plasma membrane of the ovum. The outer to the vitelline membrane is the jelly coat or albumin secreted by the walls of the oviduct. So you can see here this is the vitelline membrane, and outer to that there is a thick membrane which is composed of a jelly-like albumin which is secreted by the oviduct. Since the fertilization is external in frogs, so as soon as the egg reaches the water, the jelly coat swells up by imbibition of water, and it protects the egg from injury and against infections by bacteria and other microorganisms as well. So since this ovum is released to the external environment where there will be no immune response, and there will be a lot of pathogens, lot of disease causing bacteria will be waiting to destroy this ovum. Hence, in order to protect the ovum from all these kinds of pathogens that probably may be present in the external environment, the ovum has a very thick jelly-like albumin, which upon taking up water, so when it is expelled in the water, it takes up water by imbibition. Imbibition is the same process that you can observe in the wooden window panes as well as in the wooden doors during rainy seasons. So they take up the water and swells up. The frog's egg exhibits a well-developed polarity and the radial symmetry. The cytoplasm has two regions, the cortex and the endoplasm. So if you look at the ovum of the frog, it exhibits radial symmetry, and the cytoplasm of the ovum of the frog is composed of two regions, the outermost cortex and the innermost endoplasm. The egg cortex is a jelly-like viscous layer of the cytoplasm adherent to the plasma membrane. It is also called ectoplasm or egg cortex. You can see here, this is the ectoplasm or the egg cytoplasmic cortex of the ovum of the frog, which lies just beneath the plasma membrane. It possesses some membrane-bound spherical bodies called cortical granules containing some acid mucopolysaccharides. So this cytoplasm, the peripheral cytoplasm, which is called egg cortex or ectoplasm, is composed of numerous membrane-bound spherical bodies called cortical granules and these are responsible for the generation of cortical membrane or fertilization membrane in order to avoid polyspermy immediately after the fertilization. These cortical granules remain arranged in layers close to the plasma membrane. The dark brown pigment granules are present in the egg cortex and the animal hemisphere. So this is the special structure, special kind of pigmentation that can be observed in amphibian oocytes. The animal pole of the amphibian oocytes will be having certain darkly pigmented granules. The presence of these granules imparts the brown color to the entire animal hemisphere. The vegetal pole is whitish with slight pigment granules or little pigment granules. So this part, the animal pole part of the amphibian oocyte, will be slightly brownish in color. The reason is because of the presence of the brown colored cortical granules. And the vegetal pole of the amphibian oocyte is whitish in color, and very little quantity of pigment granules will be present in the vegetal pole. The cortical layer of the egg is stable and it is not shifted by streaming movement of cytoplasm or centrifugation force. It plays a very important role in the development of egg. The egg cortex is also responsible for establishing polarity, bilateral symmetry, and general organization of the developing egg. The details of this will be studied in the 34th session of the same paper. You can refer the session again for the study of the importance of egg cortex in the process of amphibian development. Here we are not going to study them in detail because our focus is only on the structure of amphibian oocyte in this session, not about the detailed function of it and detailed involvement of the egg cortex in the process of development. And this is a photograph of the amphibian egg in water. The endoplasm of the amphibian egg. The inner ooplasm or the inner cytoplasm of the amphibian oocyte with its nucleus is called endoplasm, which is colloidal in nature. This portion contains cell organelles like mitochondria and ribosomes, and also organic and many other inorganic substances. Endoplasm also contains a cup-shaped mass of white yolk platelets called vitelline cupola. The germinal vesicle or nucleus is located near the animal pole. The yolk granules are little and small sized in the animal pole, while they are heavily deposited in the vegetal pole. The yolk granules, it means yolk is present in little quantity in the animal pole, and its quantity is very heavy in the vegetal pole. The frog's egg is said to be mesolecital and moderately telolecital, since it contains a moderate amount of yolk which is distributed unevenly in the cytoplasm, with the vegetal pole having highest concentration. So detailed classification of ovum, including the amphibian ovum, have been discussed in the 13th and 14th session of the same paper. A detailed description of mesolithal and telolithal egg has been provided in this session. So for any further information of that these terminologies, you can go back and refer the 13th and 14th session of the same paper. So next, let us move on to the structure of the spermatozoa of cock, that means the poultry. A mature sperm of the cock is an elongated filamentous structure composed of head, a middle piece, and the tail. The head containing nucleus is drawn at the tip into an acrosome into which the Golgi bodies are accumulated. It helps in piercing through the vitelline membrane of the ovum during fertilization. The middle piece contains mitochondria which provide energy for beating of tail. The tail is extremely long, flagellum-like, and an axial filament runs throughout its length. Let us now look into the details of each and every part of the spermatozoa of the cock. The acrosome of the poultry is well defined as a separate part of the spermatozoa. As you can see here, this is sitting just like a cap over the head of the spermatozoa and is composed of the acrosome cap and an acrosomal spine. So this structure, which is almost looking like a cap, is the cap of the spermatozoa of the cock, and this part, elongated structure, you can see, and this is called an acrosomal spine. The acrosome cap appears homogeneous in structure, but the spine appears to be composed of densely packed, longitudinally arranged layered material, slightly more electron dense than the substance of the cap. Head of the spermatozoa of cock. The head is slightly curved, filiform, and is surrounded by both a double nuclear membrane and a cytoplasmic membrane. You can see here on the outside there is a cytoplasmic membrane, and inside it is covered by a double nuclear membrane. In the freshly ejaculated spermatozoa, the latter is not closely applied to the nuclear membrane along its entire length, but bulges periodically. Resulting gaps appear to be filled with the granular material. So initially, when the development is going on, that means immediately after the ejaculation of the spermatozoa, the cytoplasmic membrane of the head, it will be something like this. Okay. It will be like this. Head of the cock will be like this. Okay. Above you will find a cap of acrosome like this, and the axis of lysosome, sorry, the acrosome. Fine, a spine of the acrosome will be there. And this part is the head now. Externally, it will have a cytoplasmic membrane, and internally, it is composed of a double nuclear membrane. So, okay, nuclear membrane will be two in number, double nuclear membrane. So, this is how the structure will be. There will be no such big gaps, but anyhow, there will be a little gap. Okay. So immediately, if you look at the spermatozoa immediately after ejaculation, then you can see these two are not closely applied to the nuclear membrane. Okay, there will be a little space, and gradually, what happens? Some bulges will appear in the membranes like this, which is filled with some granular material. This observation may not support an earlier suggestion that remnants of the spermatocytoplasm appear to cover the freshly ejaculated spermatozoa. The tail of the spermatozoa can be divided into four regions here: the neck, middle piece, principal piece, and very short end piece. The spermatozoa of the mammal, in the spermatozoa of the mammal, the neck is often defined as the constricted region between the head and the beginning of the helix of mitochondria in the middle piece, and is formed by the fusion of 9 conspicuous segmented columns which merge caudally with 9 outermost dense fibers surrounding the axial filament complex. But in contrast to that, the neck of the spermatozoa of the fowl is short and composed of a complex centriolar structure from which 11 axial fibers of the tail arise. The spermatozoa of the fowl is comparatively smaller when you compare it with the spermatozoa of the mammals. And in contrast to the development of flagellum in the mammalian spermatozoa, here the neck will be composed of a complex centriolar structure. Okay, here like this will be centriolar structure having some arrangements here, and they will produce 11 axial fibers. Okay, like this. How many total? 11. They arise from this centriolar complex. Okay, and this will be present in the neck here, neck region of the spermatozoa of the fowl. So after the study of the spermatozoa of the fowl, let us move on to the study of structure of hen's egg. The hen's egg is about 3 centimeters in diameter and is polylacethal in nature. Polylacethal means it is composed of huge amount of yolk, as you can see here. The active region of development is confined to a small part here called blastodisc, and all the remaining part is completely filled with albumin and the yolk. And such eggs are called polylacethal egg because they contain a lot quantity of yolk in it. It is entirely filled up by the yolk, and over it in the animal pole lies a small cytoplasmic disc with a nucleus, and this is the active region. You can see here a small disc-like structure is present, and this is the one which gives rise to the whole embryo in the future. So this disc is called blastodisc. The yolk has a central mass of white yolk, around which are alternate concentric layers of yellow and white yolk. So if you look at the yolk, at the center you can see there is a lot of white yolk. Okay, centrally we can find a mass of white yolk, surrounding which you can see the concentrically arranged yellow and white yolk. So they produce concentric layers of yellow and white yolk. The central flask-shaped white yolk is called vertebra and runs from the center of the lower side of the blastocyst, and there it spreads to form the nucleus of Pander. You can see here this structure, the central yolky mass, which is composed of white yolk granules, or a flask-shaped structure. Okay, and this structure is called vertebra. And this vertebra runs from the center, from here, to the lower side of the blastocyst. So this is the blastocyst, this is the lower side of the blastocyst. So from the center, the vertebra moves towards the lower side of the blastocyst, and there it spreads to form some structure called nucleus of Pander. And just below that there will be a small connection which is called the neck of vertebra. The yolk and blastocyst are bounded by a plasma membrane and outer vitelline membrane, which is converted from zona radiata. There will be a plasma membrane. Okay, there will be a plasma membrane, and outer to the plasma membrane there will be a vitelline membrane. Okay, there will be a vitelline membrane. A plasma membrane will cover the entire structure, including the blastocyst. Okay, from here, and immediately exterior to this plasma membrane there will be one more membrane called the vitelline membrane, which is formed after the conversion of zona radiata, the outermost layer of the ovum. The vitelline membrane is of double origin. The inner layer of this membrane is produced in the ovary between the oocyte and the follicular cells and is composed of very tough fibers. The outer layer is formed in the upper part of the fallopian tube. So the outer and inner vitelline membrane has two different origins. The inner layer is formed from the ovary, and the outer layer is produced from the fallopian tube, while the egg is passing through the fallopian tube before it is being laid. The yolk, you can see here, the yellow structure is the yolk. And this yolk is highly nutritious and provides all the required nutrients for the developing embryo during the development. This yolk contains about 48.7% of water, 32.6% of phospholipids and fats, around 16% of proteins, and around 1% of carbohydrate and 1.1% of other chemical molecules and nutrients required for the normal growth and development of the embryo. It also contains proteins, and these proteins are in the form of phosphatin and lipovitamin or levatin. The fat present in the egg is a neutral fat. About 50% of it is the neutral fat, and rest of it is the phosphated cerebrosides and the cholesterol. The fertilized egg or zygote is covered by a layer of dense viscous albumin, which forms a thin chelaziferous layer around the vitelline membrane. So this is the yolk. You will have a plasma membrane, and the plasma membrane will be surrounded by a vitelline membrane. And outer to the vitelline membrane, you can see the entire structure is covered by a proteinaceous substance called albumin. And this dense albumin forms two twisted cards, as you can see here on either sides. And these cards are chelazae, which are present on both the sides of the vitelline membrane, this side and this side as well. So these chelazae, which is formed out of dense albumin, they form two twisted cards or chelazae, one at each end of the zygote. They are formed by rotation of egg during its movement through the oviduct. See, immediately after the vitelline membrane, there will be a very thin structure called chelaziferous layer, which is composed of dense viscous albumin. And on either side of the yolky mass, this dense albumin forms two twisted cords or chelazae. These structures are called chelazae, and this twisting takes place during the movement of the ovum in the oviduct. So there will be some rotations which leads to their twisting of the chelaza or in the yolky mass. So around the chelaziferous layer is a thick layer of watery albumin. So this structure, the vitelline membrane, will be covered by a chelaziferous layer, and exterior to the chelaziferous layer, the entire space will be filled by a thick layer of watery albumin. Albumin will be watery in nature. All albumin is secreted by the upper glandular walls or magnum of the oviduct. So the entire albumin is secreted by the glandular walls of the oviduct. The function of the albumin are to provide nutrition to the embryo, and also serves as a store of water, and also acts as a protective envelope for the developing embryo, and it protects the embryo from mechanical and chemical injuries during the development of the embryo. So this is the yolk, and surrounding the yolk of whitish fluid, you can see that is the albumin. The albumin and the yolk also contain a variety of enzymes, vitamins, pigments, and phosphorus. The isthmus part of the oviduct secretes two shell membranes made of tough keratin fibers matted together. The two shell membranes are closely applied except at the blunt end of the egg, where they are separated by an air space formed after the egg is laid. The nidamental glands of the oviduct secrete a porous calcareous shell which soon hardens. So surrounding this yolk and the albumin, two shell membranes are secreted by the isthmus part of the oviduct. And this structure is tough because of the presence of keratin fibers that are matched together. And these two membranes which surrounds this yolky mass as well as the albumin are opposed together. They are closely applied together so that a form structure is established. And when the egg is passing through the oviduct again, the nidamental glands of the oviduct will secrete a calcareous shell which is porous in nature and will soon harden after the egg is being laid. You can see here, this is the photograph of an egg containing numerous holes here. The porous shell of the ovum can be observed here. So all the lighter patches here are the pores. Hence the external surface or the wall of the ovum is highly porous. And the calcareous shell is pierced by a large number. It means about 7,000 of fine pores filled with a protein related to collagen. The diameter of the pores varies from 0.04 to 0.05 millimeters. These pores allow exchange of gases, mainly oxygen and carbon dioxide, during the respiration of the developing embryo. The egg is laid 24 hours after the fertilization, and its further development takes place when the egg is incubated by the female. The incubation must continue steadily for 21 days at the temperature of 103 Fahrenheit. That's all about the structure of the spermatozoa of frog and the fowl, and the structure of ovum of the frog and the fowl. For any further information about these topics, I would recommend you to follow these textbooks. Thank you.